My name is Amanda Larracuente, and I'm in the Department of Biology at the University of Rochester. And my lab is interested in sex chromosome evolution. And so as Thielen described so nicely, our classic view of sex chromosome evolution is that sex chromosomes evolve from a homologous pair of autosomes, where one chromosome acquires a sex-determining locus and suppresses recombination around this locus, and then these chromosomes begin to differentiate in this region, ultimately leading to heteromorphic sex chromosomes. And in species where males are the heterogametic sex, we have a Y chromosome down here as the degenerated chromosome, and the X chromosome is not. So sex chromosomes have evolved independently many times, and old Y chromosomes share some of the same properties. And so they tend to be, they're male restricted, they tend to be non-recombining over most, if not all of their length, and as a consequence of this, they tend to degenerate and accumulate lots of repetitive sequences and become heterochromatic, which means that they're silenced in most cell types and tissues. And so this is a pair of human sex chromosomes showing that the Y chromosome is really small compared to the X. But my lab studies Drosophila sex chromosomes, and they are different in a number of ways. But they also share some of the same properties. So in fruit flies or Drosophila, the Y chromosome is not sex determining, but it is required for male fertility. So males that, or flies that don't have a Y chromosome are males that are sterile, but otherwise pretty normal. So the Y chromosome is about 40 million bases long, and it has a long and a short arm, and it only has about 20 genes on it. And so these 20 genes are spread across 40 megabases, so this is a really low gene density on this chromosome. And what's interesting about these genes is that they're all acquired from the autosomes. They all move from somewhere else in the genome to the Y chromosome. And 80% of this chromosome is really dense and simple satellite repeats. So it's a very heterochromatic chromosome. So satellite DNAs are these large blocks of tandem repeats that make up the bulk of heterochromatin. They tend to occur near centromeres or telomeres and on Y chromosomes, and they can occupy hundreds of kilobases or even megabases of sequence on chromosomes. And so we understand Y chromosomes in flies at the cytological level and to some extent at the genetic level, but at the base pair level, so sequencing level, it's really difficult to understand Drosophila Y chromosomes because they're so rich in these satellite DNA sequences. And so the reason is because repeats are really difficult to sequence. There are a number of reasons for this that if you're around this afternoon, I'll tell you a little bit more about this. But they end up being underrepresented in sequencing reads when you have a sequencing project. And then even if you have the reads from these regions, they're really difficult to assemble into a contiguous bit of sequence. And so if you imagine you have this red repetitive sequence read, you don't know where it goes in the genome unless it overlaps something that's unique. And so our traditional sequencing technologies have short read lengths. They can't span repeats, and it's very difficult to assemble repetitive regions of the genome. So our lab is using single molecule real-time sequencing from Pacific Biosciences to circumvent at least some of the problems that we have with traditional sequencing methods. So this is a long read sequencing technology. The average read length is about 15 kb, depending on which platform and when you do your sequencing, but they can get up to 60 kb long. And so these long reads span repeats and lead to better genome assemblies in general. So what I want to tell you about in this talk today is the work that we've been doing with our PacBio genomes to look at Y chromosome assemblies, or at least start to attempt to assemble some of the Y chromosomes in these old Y chromosomes in Drosophila species, and then what we've learned about Y-linked genes from these assemblies. So our species that we work on is the famous Drosophila melanogaster. This is our model organism that geneticists love. And then we also look at a closely related clade of species called this in the simulans clade. So this is Drosophila simulans, mauritiana, and sechellia. So the simulans clade, the species in the simulans clade have a common ancestor about 240,000 years ago, and then this clade diverged from melanogaster about 2 million years ago. So these are all really closely related species, and if we're able to look at Y chromosomes in each of these species, then we can look at Y chromosome dynamics over a very short evolutionary timescale. So So in Drosophila melanogaster, Pacific Biosciences, the company, sequenced melanogaster to about 95x coverage with PacBio sequencing. They did this in collaboration with the members of the fly community. So we used these data. And then our group, in collaboration with other groups, mainly at UC Irvine and a couple other universities in this country, sequenced the genomes of simulans, mauritiana, and sechellia between 85x coverage to 120x coverage. So this is really deep PacBio sequencing. And I'm not going to talk about the details of how we build these assemblies in this talk today, but the general idea is we take these PacBio reads, we create de novo assemblies of the genome. Now, each of these genomes already has a reference genome assembly, and they were made with short reads. Okay, and so some of them are better than others. So melanogaster is actually a very good genome, but the other species are not. And so we're building new reference genomes with the PacBio sequences. But one thing that we were sort of surprised to find is that with our standard PacBio assembly methods, we don't get the Y chromosome coming together in contigs at the other end. And so this is a little bit disappointing, but we have to take extra measures to go and assemble the Y chromosome. And so I'm not going to tell you exactly what we do now. If you're around this afternoon, I'll tell you more about our methods. But the general idea is after we build these de novo reference assemblies, the new ones with PacBio reads, we take the PacBio reads again and we map them to these genomes. And then we take out all the reads that we know came from the X chromosome and the autosomes. And so what we want to do is we want to enrich for Y-linked reads. And so if we throw out those reads, we enrich for things on the Y chromosome, but we're also ending up pulling out some of these reads that map to other heterochromatic regions of the genome. That's okay. And then we take those leftover reads and then we assemble those. After that, we do a lot of manual curation and we confirm Y-linked contigs with short read data in males and females. Okay, so before I tell you what these Y chromosome assemblies look like, let me tell you where we were before we started this. So this is the Y chromosome in the melanogaster reference. This was just released last year. This genome is pretty spectacular, actually. And the Y chromosome is, we have about 4 megabases assigned to the Y in the species. And for this, I mean, this is pretty remarkable in itself. So this is the reference genome that we did not do this. But it's split into 261 contigs, so that's a lot of fragments. And the N50 size is over 81 kb. So our PacBio genome for melanogaster, we increase the amount that we can assign to the Y chromosome to 15 megabases, or over 15 megabases. And it's on fewer contigs, so we improve our contiguity. And so this means that on average, 415, our Y chromosome is contained half on 415 kilobases or larger. So we have big pieces coming from this Y chromosome. Now, if we look at our simulans genomes, my PowerPoint quit. So if we look at our simulans genomes, we actually do a bit better. So when we had this, when we made this reference, at the time, the simulans clade genome references that were out there were not actually, there was no Y chromosome assembled. And so we were pretty much starting from scratch in these species. So anything that we get from the Y is an improvement on what we had currently. Okay, so let me show you what that looks like. We're actually doing a little bit better in terms of contiguity than melanogaster. So we have 14 megabases assigned to the Y chromosome, and this is in fewer contigs, and our M50s are a lot better. And so what this means is that we have 30 to 40% of each of these Y chromosomes in contigs in each of these species. So this is pretty exciting. Okay, so now that we have a lot of the Y chromosome in contigs, we can start to compare them and look at evolution of Y-linked genes. And so as I told you before, the Y chromosome acquires genes from other places in the genome. So most of these genes jump from autosomes onto the Y chromosome. So here's a list of a lot of melanogaster. This is not all of them. The melanogaster Y-linked genes and where they come from in the genome. And here's a list of their putative function. So we don't really know what most of these genes do. We have an idea that they do something in spermatogenesis. Many of them seem to function after meiosis in spermatogenesis, but we don't really know a whole lot about these genes. But we can look for these genes in our simulans, clade, Y chromosome assemblies and ask if they're there. Okay, and so when we do that, we do find most of these genes are in these assemblies, except for these three down here. And these three genes are known to be melanogaster specific, so they jumped onto the Y chromosome in the melanogaster branch. And actually, all of this information on this slide we knew from the work of Bernardo Carvalho and Andy Clark. They've spent a lot of years doing really beautiful work annotating genes on the melanogaster Y and asking if they're in these other species. So this is we're using this as a way to say that our assemblies are recovering all of these genes. But now what we can do is we can look at the order and the orientation of each of these genes to study gene organization on these chromosomes, which is something that we could not do before. And so here I'm showing you the melanogaster Y-linked genes. And so each gene has a different color, and I'm indicating the orientation with the arrow. Okay, so I'll just show you mauritiana to start. So this is our best guess at the order and orientation on the mauritiana Y chromosome, and what you can see is there's a lot of evidence for gene rearrangement. So, for example, KL3 and KL2 are adjacent on the melanogaster Y chromosome, but they are not in the mauritiana Y chromosome. It looks like there's been an inversion because we have a different orientation, and then there are a bunch of genes that are now in between them. And so we see a lot of evidence for rearrangements of genes. And so we can look now within those closely related species and ask how much gene rearrangement do we see, even on this short evolutionary timescale. And the answer is that we see a lot. We still see genes that are adjacent in one species being flipped and split apart in other species. And so over short evolutionary timescales, the genes are really shuffling on these Y chromosomes. Okay, we also find lots of evidence for gene duplication. And so, in fact, most of the genes on the Y chromosome that we identified have exons that are present in duplicate copies. A lot of these duplicate copies are degenerated, which in this case, we're calling something degenerated if it has a deletion in it. And so here's an example of a gene called ARY. This is a small gene. On the x-axis, I'm showing you it has 6 exons. This is melanogaster, mauritiana, simulans, and sechellia. And on the y-axis are the copy number. And so this exists as a single copy gene in melanogaster and mauritiana, and 2 copies in simulans and sechellia, and then it has extra exons that are degenerated. So here's another example of, this time, a very large gene, KL2. It has twice as many exons, and it's present in a single copy. This gene is spread across a very large part of the Y chromosome, and so it only has partial duplications. So everything that we know about the Y chromosome in these species is melanogaster-centric. It's from what we know about melanogaster. And so now that we have these assemblies, we can look for new genes in these other species and get a different perspective. And so here we're looking for genes that jumped onto the Y chromosome in the ancestor of the simulans clade. So we find evidence right now, at least, for 4 independent duplications. So this first one is CG3511. This is a gene that we think jumped onto the mauritiana Y chromosome from chromosome 2R. And so this is another gene that was acquired from an autosome. Now, this is the gene structure of the parent gene, which is still on 2R in mauritiana. So these boxes are the exons. And this is a multi-copy gene on the Y chromosome. I'm showing you 3 of the copies here, and all 3 of them appear to be inactivated. And so they've accumulated. These gray bars are showing you where the deletions are, and there's insertion of transposable elements. And so here's a gene that jumped onto the Y chromosome in the mauritiana branch and then degenerated really rapidly. So similarly, HRB27C is also an inactivated gene, except for this one moved from chromosome 2L onto the Y chromosome of the sim clade ancestor. And so it's present in all 3 species, but it appears to be inactivated. CG16781 is interesting because it jumped onto the Y chromosome, this time from the X chromosome in the simulans clade ancestor. And it appears to be degenerated in simulans and sechellia, but it's still active in mauritiana. And then this last gene, SRPK, is the one that we're most excited about. is present in many copies on the Y chromosome of all 3 species, and it appears to be active. And so if you're sticking around later, I'll tell you a little bit more about what we've learned about this gene and why we're excited about it. And so we're really interested in these new genes that are born on these Y chromosomes and how they change how spermatogenesis is wired in each of these species. And it'd be interesting if there are species differences that contributed to incompatibilities and things. And so one candidate for an interesting gene that is now has a function in spermatogenesis that's coming from the Y chromosome is SRPK. So after meiosis and spermatogenesis, these newly haploid spermatid have to shrink their nuclei to fit inside the needle-shaped head of the sperm. And this is at least in part accomplished by swapping out histones for these small histone-like proteins called protamines. SRPK in human phosphorylates histones and facilitates in this histone-protamine transition. And so this is an exciting gene because this is now duplicated to these Y chromosomes and is present in many copies. And so it might be doing this in facilitating this transition in the simulans clade species now from the Y. Okay, to summarize, we've used PacBio sequencing to assemble 30 to 40% of these Y chromosomes in melanogaster and the simulans clade species. And we find lots of evidence for reorganization of the genes on these Y chromosomes, gene duplications, and lineage-specific genes that are still active. And so with that, I'd like to thank Ching-Ho Cheng. He's the one who did all of this work, and he's here. And if you are sticking around later, you'll get to hear him talk about a different Y chromosome. Casey Bergman is someone who helped us get set up with PacBio sequence or PacBio assembly and melanogaster. We collaborate with J.J. Emerson and Christy Montooth, Colin Mickeljohn, and Jeffrey Venenaikum on SimClade PacBio genomes, and Mehul Chakrabarty with Ching-Ho did a lot of the initial genome assembly to build the reference genomes.